Hello, everyone. My name is David, and I'll be presenting Nipah virus along with Esteban, Megan, and Amira. The focus question for this presentation that we will answer are what molecular factors lead to the polytropism of Nipah virus? And here's the definition of viral tropism right down below, which is the ability of a virus to infect particular specific cells, tissues, or host species. So some base facts about Nipah virus. Has a 40 to 80% fatality rate. It's a negative single-stranded RNA virus that's non-segmented. The scientific name for it is Henipavirus hence. The genus is Henipavirus, and there's a list of a few viruses that are also included in that genus. The family is Paramyxoviridae, with its closest relative being Hendra. There's two main strands of Nipah virus, NIV Malaysia and NIV Bangladesh, which I will go into more detail on the next slide. The reservoir species for this virus is the spectral flying fox, but it's also found in pigs, horses, dogs, and different species of bats. The main method of spread for this virus is bodily fluids and consumption of infected meat. And it's labeled as a category C virus with the biosafety level 4. And to the left right here, we have some images of Nipah virus under the microscope, with figure A being a single nucleocapsid of Nipah virus, B being viral nucleocapsids of the virus and cross longitudinal sections. And finally, section C is where the virus particle is with nucleocapsids enclosed in viral envelope. So, going into our outbreak timeline, this virus originated in Malaysia and was named after its original place in Malaysia, which was Sungai Nipah, Malaysian village. And it was first found in infected pigs on pig farms, and the mainly infected pig farmers are farmers, when they noticed that there was an outbreak of this illness, they decided to try to control it by having extra sanitation at the farms and culling infected pigs and making sure to keep an eye on it over time. The main, most dangerous outbreak was in 1999. And in Singapore, there was this outbreak where they were handling infected pigs again. And in 2001, there were two outbreaks, one in India, with another one in Bangladesh. And this was the start of a long run of outbreaks for Bangladesh, especially, with multiple different outbreaks occurring throughout the years, all the way up to the most recent one in 2023. Whereas in India, the outbreaks occurred due to infected bats. And in the Philippines, there was outbreaks over there in 2014, but we didn't know at the time that it was Nipah virus until a few years later when it was researched what this outbreak all was about. And in 2018, the Nipah virus outbreaks in India continued for 2018, 2021, and 2023.
Just so we're on the same page, let me give you a brief rundown on how Nipah virus replicates. Nipah, as an envelope virus, enters the host through fusion. This is done by the virus attaching to cells, leading to binding with efferent receptors, where it then releases its content into the cytoplasm. The content that is released is the usual content expected for a negative strand RNA virus, being RDRP, and the viral genome being the most vital components released. RNA is duplicated in the host cell, leading to the production of 9 proteins. After creating a considerable amount of N proteins, the N protein wraps around the viral genome as they are being created, leading to the production of complete viral genomes. With the expression of M protein, the virus will then lead its component to the cellular membrane for viral assembly and eventual budding. Another crucial detail to know is the function of the proteins and how the proteins are created. As mentioned earlier, there are 6 structural proteins and 3 accessory proteins created through gene editing. Some proteins are self-explanatory, with fusion proteins being used during fusion, and glycoproteins, which help with attachment and recognition of efferent receptors on the host. Some other proteins have obvious functions, but also some unique functions, like large RDRP having PRNTase and MTase functions for mRNA capping. Matrix proteins form the shell of the virus but also function as a shuttle that allows virus proteins to enter the nucleus. The NIP proteins are used for genome replication, and finally, the accessory proteins C, V, and W are used for immune antagonism. Some of these accessory proteins are created from the polymerase stuttering at the gene editing site, adding extra Gs, creating a frameshift dictating the production of V, W, and P proteins. Finally, C proteins have an alternative start codon leading to a reading frameshift. The accessory proteins W, V, and C, alongside proteins P, have some form of immune antagonism in the JAK-STAT pathway. The first form of inhibition we see is V proteins binding to Reg I and MDA5 to stop phosphorylation or activation of these proteins, blocking the production of IF and beta. We also see W proteins inhibiting the TLR3 and viral RNA inducting signaling pathway, which is done by binding to TBK1 and IKKE proteins to destabilize IRF3 proteins. V proteins can mimic this function in a similar manner but can only bind to TBK1. Lastly, we see protein V and P inhibiting STAT1 phosphorylation in the cytoplasm and protein W in the nucleus. V proteins can also do this to STAT2 proteins. While there is no definitive function for C proteins, it is believed to either bind to IKKA to inhibit IRF7, similar to W and V protein does to IRF3 protein, or it limits viral protein synthesis to lessen the chance of detection. Antagonism of the JAK-STAT pathway can provide one reason why we see a great deal of polytropism in Nipah virus. The STAT protein family is a highly conserved protein family within mammalian hosts. We see a wide range of mammals ranging from humans to mice having some form of JAK-STAT pathway. Not only is the STAT protein highly conserved in mammalian hosts, but it's also conserved in wide range of tissue cells. In one of our previous lectures, Lecture 8, we talk about how specifically type 1 IFN are found in all nucleated cells, type 3 IFN are found on natural killer cells, and type 2 are found on epithelial cells. These type 1 and 3 use STAT1 and 2 proteins, while type 2 use only STAT1 protein. This is one potential reason why we see polytropism in the Nipah virus.
Right here are images of the entry mechanisms. And the viral glycoprotein G binds to two highly conserved host cell receptors, Ephrin B2 and Ephrin B3. And these two host cell receptors are found throughout the body, such as the digestive system, urinary system, and cardiovascular tissues. And because of how apparent they are throughout the body, this allows the virus to establish infection in multiple different places, which allows it to have such remarkable polytropism. Once the G protein binds, it triggers a conformational change that exposes the fusion protein F, and this fusion protein connects with the host cell membrane. And once it connects, it allows the viral genome to enter the cytoplasm. And the coordination between G and F proteins create a highly efficient entry system that gives Nipah a significant advantage over other related paramyxoviruses.
Now that we understand how Nipah is able to infect a variety of cells, we can understand the pathology of Nipah virus. The highly conserved receptor Nipah targets allows it to infect the endothelial cells that make up our vascular system. The circulation of blood then spreads the virus to systems and tissues throughout the body. We know highly vasculated regions like the kidneys are impacted, but the most clinically significant and consistently impacted systems are the pulmonary and central nervous systems. The pulmonary system is impacted because it is a highly vasculated region susceptible to infections and tends to be the site of entry for the virus. The central nervous system being impacted requires the infiltration of infected immune cells, the virus traveling through the olfactory nerves in the back of our nose directly to our brain, or by compromising the blood-brain barrier, which is the layer of cells that generally prevents contents from the blood entering into our cerebral space. The disruption of the blood-brain barrier can be the result of progression of infection. After the 3 to 14-day latency, individuals infected with Nipah virus experience nonspecific symptoms like fever, headache, coughing, and sore throat. This is why Nipah is often misdiagnosed, as it was in the 1999 Malaysia outbreak. However, as the virus continues to damage vascular tissues, it weakens the blood-brain barrier, allowing infection of the brain. More importantly, the immune system responds by upregulating inflammatory cytokines. This inflammation in the lung and brain is responsible for the more severe complications from Nipah virus infection and only further compromises the integrity of the blood-brain barrier. Pulmonary complications of Nipah virus include pneumonia, pulmonary embolism, and acute respiratory distress syndrome, or ARDS. On the right, you can see a comparison between a normal chest x-ray and the x-ray of a patient from the 2004 Faridapur, Bangladesh outbreak. As you can see, there is a white cloud, which is the fluid within the lungs, the result of leaky veins and inflammation. Central nervous system complications include brainstem dysfunction, coma, and encephalitis. Again, on the right, you can see a comparison between a normal head MRI and an MRI from a patient from the 1999 Malaysia outbreak. The little spots you see are lesions. And while the patient did not die from her acute Nipah encephalitis, she, along with several other patients in this study, experienced long-term neurological symptoms. Survivors and families of survivors reported long-term personality changes, chronic fatigue, depression, and verbal and visual impairments. While the severity of acute and long-term symptoms vary with each strain, They often progress rapidly, with doctors from the 1999 Malaysia outbreak reporting patients going from stable to comatose in a matter of hours. Unfortunately, there are no approved vaccines or therapeutics for Nipah virus infection currently. The antiviral drug ribavirin was shown to reduce mortality in the 1999 Malaysia outbreak, but the mechanism in which that drug disrupts Nipah virus is not understood. Two other drugs, VV116 and favipiravir, have been shown to reduce mortality in hamsters. On the right, you can see graphs depicting log RNA genome count in lung, brain, and spleen tissues decreasing in response to the drug. However, once again, the mechanisms of these drugs, and specifically how they interact with Nipah virus, are not understood.
So one of the most interesting unanswered questions surrounding Nipah virus is why bats can carry the virus without developing severe disease. As shown here, Terapus fruit bats are the natural reservoir hosts, meaning the virus can persist and circulate within bat populations over time without causing widespread disease. In humans, however, Nipah infection can cause severe respiratory disease, encephalitis, and mortality rates that can exceed over 70%. Research over the past decade suggests that bats may maintain a unique antiviral state. For example, several studies have showed elevated baseline type 1 interferon signaling, meaning some antiviral pathways are already active before active infection occurs. This could help limit viral replication early. Bats also appear to regulate inflammation differently than other mammals. In many severe viral infections, including Nipah itself, much of the pathology comes from the host's inflammatory response rather than the virus alone. Bats seem to be able to control the virus while avoiding excessive immune-mediated tissue damage. However, it's really important to emphasize and recognize that this is still an active area of research. We have several hypotheses, but there's currently no single accepted mechanism that completely explains why bats tolerate Nipah virus infection while humans and other mammals develop severe disease. Understanding this, however, could potentially reveal new therapeutic strategies for reducing disease severity in humans. So, although the bat reservoir question remains an active area of research, the evidence we've discussed throughout this presentation provides a much clearer answer to our original focus question: what molecular factors contribute to Nipah virus polytropism? Our findings suggest that Nipah virus polytropism is driven by a combination of conserved receptor usage, broad receptor distribution, and immune evasion. Nipah virus uses FNB2 and FNB3 as entry receptors, which are highly conserved across mammalian species. This allows the virus to infect a wide range of hosts without requiring major adaptations. Because these receptors are expressed in multiple tissues, including respiratory, endothelial, and nervous tissues, Nipah gains access to several organ systems within a host. Once infection reaches the vasculature, the virus can disseminate throughout the body. Combined with immune evasion mechanisms mediated by VWNC proteins, this enables widespread infection and severe disease. Taken together, these mechanisms suggest that Nipah's polytropism is not driven by a single factor, but by the combination of conserved receptor usage, broad receptor distribution, and effective immune evasion. Together, they provide a molecular explanation for how Nipah virus is able to cross species barriers, infect multiple tissues, and cause disease. While Nipah outbreaks are typically limited in size, the virus continues to reemerge across multiple countries and is associated with high mortality rates. Repeated zoonotic spillover events, documented human-to-human transmission, and the absence of approved therapeutics continue to make Nipah a really important public health concern. Despite these advances, important questions remain. While we now understand many of the mechanisms that allow Nipah virus to enter cells and affect diverse tissues, much less is known about what determines disease severity once infection is established in different organs. For example, why do tissues experience more severe damage than other, and how do tissue-specific responses contribute to respiratory disease, encephalitis, and systemic pathology? Answering these questions could help identify more precise therapeutic targets and improve our ability to respond to future outbreaks. Because of these characteristics, Nipah has been designated a WHO priority pathogen and remains a major focus of emerging infectious disease research. Ultimately, understanding the molecular basis of Nipah polytropism provides a foundation for improving surveillance, developing future therapeutics, and preparing for future outbreaks.
And here are our sources.